This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV. This Week in Virology, episode 455, recorded on August 18th, 2017. This episode is brought to you by the Defense Threat Reduction Agency, part of the U.S. Department of Defense. The agency's Chemical and Biological Technologies Department hosts the 2017 Chemical and Biological Defense Science and Technology Conference to exchange information on the latest and most dynamic developments for countering chemical and biological weapons of mass destruction. Find out more at cbdstconference.com. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining us today from southeastern Michigan, Kathy Spindler.
Hi, everybody. It's 75 degrees Fahrenheit, and that's 24 Celsius, so it's just about perfect. It's partly cloudy, but more importantly, it's partly sunny.
Here it is 27 Celsius and cloudy. It was raining this morning. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
Well, welcome back.
Thank you very much. It's a shocking 94 degrees here, on its way, according to the forecast, to 101, but where the humidity is a mere 44%. 2.69 degrees. So, it's okay. Beautiful blue sunny sky.
You're back from your travels?
Back from our travels. We got back on Sunday.
And you were gone for over a month, weren't you?
We were gone for almost 2 months.
Last week, I think you were in Yellowstone, or was it the week before?
No, it was last week I was in Yellowstone. The week before you were on the road, so we didn't record. Week before that, what it said I was busy, right?
Yeah, probably sailing or something.
There was some. Oh, I know. Yeah, I sent you pictures from the Oregon coast. Mm-hmm. My wife and I took a little side trip to the Oregon coast. Saw some old friends who from the University of Oregon in Eugene, and traveled up the coast a little bit. It was great.
You were in Yellowstone like a day after Dixon was.
Something like that.
So yeah. Dixon dropped me off at the airport on Wednesday, Missoula, and then he was driving down to Yellowstone, which was about 6 hours south, and he said he was going to stay there for a couple of days. So it was just funny that you were all...
Yeah, he was probably going to go fishing, right?
Yeah, no, in Yellowstone, he was just taking pictures of wildlife.
Oh,
and I think, Kathy, you were not far away either, right?
Right. I was in Glacier, and that's when I saw my grizzly bear and met the TWiV.
Oh, you actually saw a grizzly bear?
30 yards below us on the trail where we had just been.
Ooh.
Yeah.
And met a TWiV fan?
Yeah, she was hiking with me at the time.
Maybe
and two other women.
Maybe the bear was also a TWiV fan.
He was after moths. This was up in the Talus, and Dixon sent me an article that they can eat up to 40,000 moths a day. So they turn over the rocks and they eat these fat chiclets, as they're called.
Wow.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. I thought you were never going to get to me.
Yeah. Well, we're almost done anyway, so.
Yeah, it's overcast and sticky and 80 Fahrenheit, 27 C, and it keeps threatening to rain, and eventually I think it just might.
We're all over the country today, and now we have a guest from Seattle, Washington. She is at the Fred Hutchinson Cancer Research Center. Erin Garcia, welcome to TWiV.
Hi, everyone. Thanks for having me.
What's the weather like there?
I'm about to make you all really jealous, depending on your preference. It is 64 degrees Fahrenheit, 18 degrees Celsius. My weather app says cloudy, but when I look out the window, the sun is starting to make an appearance to crystal clear blue skies. So it's going to be quite a nice afternoon, I think.
64 is just way too cold.
It's the high is going to be about 70, I think, towards the end of the day, but it's pretty early right now. So
you probably don't have
it's only 10 a.m. your time, right? 10, 15?
It's 11.
Oh, 11.
So
you don't have any. You don't have any humidity, right?
Not that you can really feel. It's about 74%, but the dew point is 13% or 13 degrees. So you really can't feel the moisture in the air much.
Well, Dixon is driving south, I think, to where? Where's the best place to see this eclipse? Kentucky or something like that?
Yeah, there's a whole stripe across the country.
Depends on your longitude.
He's driving south, and they're going to see this eclipse. So he's
South Carolina, going west from there, I guess somewhere in the Kentucky, Tennessee area, southern Illinois.
I think he mentioned Oak Ridge or something like that.
Oh, okay.
There's going to be millions and millions of people there.
Oh yeah, I want to stay as far away from this thing as I can get. You know, it's going to be a full eclipse very close to where we were in Oregon, and they're expecting millions of people, and they don't know how to handle it.
Right where I am in Seattle and not going anywhere near Oregon.
Yeah,
I am one of the fools. Going to be in South Carolina, but we'll see how that goes.
Are you flying?
Yeah, we're going to fly down. Did I mention this last time?
I think so.
Yeah.
Are you flying in yourself, or are you taking?
Yes. Yeah. So we're flying Air Allen down to South Carolina. We're going to stay in the Myrtle Beach area, which is outside the zone of totality. and it's funny because if hotel rooms inside the zone of totality, you can't get one. They've been booked for months. And immediately outside it, just, Myrtle Beach is like 20 miles outside the zone or something. And it's the late summer discount season, and they've got rooms available and everything's cheap, and come stay with us. But of course, the challenge then is how do you get that 20-mile distance on roads that are going to be totally gridlocked? And I'm hoping to bypass that. So we'll see.
Oh, good luck.
Yeah, thank you. The weather forecast is still kind of iffy, so that's
the big deal. I mean, if it's cloudy.
Yeah.
So I have my eclipse glasses, so I am going to go outside and watch it here. I blocked off my calendar for no appointments between 1 and 3:45.
All right, great. Erin, where are you from originally?
I'm originally from New Mexico.
Wow.
So very different.
And
spot.
What part of New Mexico?
I grew up in the central area, so basically a suburb of Albuquerque. It's a little town called Rio Rancho. It's actually the fourth biggest city, I think, in the state, although that it may have changed in recent years, because it used to be one of the fastest growing cities in the country for a while, because Intel put a plant there. So engineers from all over the country were moving to Rio Rancho, but as it started slowing down, people have started slowly moving away. So
you said Las Lunas?
Rio Rancho.
Rio Rancho.
One of my memories of Albuquerque is going to a restaurant and seeing Bob Krug there having lunch. Bob Krug, a well-known virologist. New Mexico is a destination for many scientists. You may know there are meetings of all sorts up in Taos.
Oh, yeah.
Ski meetings people go to. Flying to Albuquerque, drive up to Taos or Santa Fe, too. There are meetings there as well. And,
one of the girls in Harmeet Malik's lab went to, and one of our staff scientists actually went to a Gordon conference or a Keystone meeting in Santa Fe. I can't remember which one it was.
Yeah, Nell Zeldy over on TWiVo. Not too long ago, we did an episode, and he was at the hotel there in Santa Fe. And did you go to high school as well in Rio Rancho?
I did, yes.
Where'd you go to college?
So I started out at the University of New Mexico, which is in Albuquerque. So it was like a 30-minute drive from my parents' house. And then I realized I was way too close to home. And so I transferred to New Mexico State University, which is in Las Cruces, New Mexico, and is about 3.5 hours south.
And you were a bio major, I guess?
I was a microbiology major.
Wow. So you went to college with the idea of being a micro major?
I did. So I actually took a microbiology class in high school that kind of piqued my interest in microbes. And so when I went to college, I knew that's what I wanted to study.
Wow, that's kind of unusual for high school, right? A micro course?
Yeah, so we actually had what I was told, and thinking back on it, I'm not entirely sure how true it was, but we were told this was a BSL-2 lab in my high school. So we had like a little room where we grew cultures and could do gram stains and
neat
that kind of stuff in that little area of the classroom. So it was awesome. And I learned a lot.
Cool. Wow.
And now you are a PhD student in the Emmerman lab, right, at Fred Hutch?
Right.
And that, now your PhD, is there an association with UW? Is that how that works? Or do you have
Yes, so I'm in the Department of Microbiology at UW, but then my home lab is at Fred Hutch. So I have dual appointments at UW and Fred Hutch.
So Rich Condit and I did a TWiV with him years ago. You probably weren't there at the time, I guess.
I was not. I was in Cleveland, Ohio doing a postbac at that time, but then I listened to it the year after when I was thinking about rotating in his lab.
Remember, Rich?
Oh, yeah. Clearly.
I think that's many people tell us they listen to the TWiVs of people they're thinking about or that gets them interested in a particular lab. So that's a good service to do.
It's really helpful because I kind of got a sense of where the lab came from and kind of the story that took Michael along his path and things that happened in the lab before I joined.
How far along are you in your work there?
I'm about to start my 4th year. So I'm heading, I would guess, kind of towards the end. I'm in the big black box where you never know what's going to happen. So,
what do you work on?
So I work on a host protein called ApoBec. So cytidine deaminase is ApoBec 3H is my protein of choice.
A weapon of mass deamination.
Exactly.
Perfect for our sponsors for this episode.
So the protein that I study, like Michael, when he was talking in his TWiV episode, he focused a lot on ApoBec 3G and a few ApoBecs in chimps that are important. But the protein that I study has had a long history in the lab where this protein is actually polymorphic in humans, where some forms or some haplotypes are active against lentiviruses, but some haplotypes are completely inactive. And this has to do with protein stability. So my project is kind of looking at the evolutionary importance of ApoBec 3H. So I look at it in a system of primates, African green monkeys, to try and figure out more from an evolutionary standpoint how important ApoBec 3H is for restricting lentiviruses such as SIV and HIV.
Do you know what you're going to do next when you finish up?
Not quite. So I'm kind of at the point where I'm thinking about what I want to do, and one option is to go on and do a postdoc, which I'm kind of thinking about maybe doing a postdoc internationally, just to kind of explore that side. Or I could, and something I'm also really thinking hard about is whether I want to get into science writing. Which has absolutely
nothing to do with developments going on in this country right now, right?
None at all.
It sounds like a smart idea in that context as well.
My husband has always said that he really would love to live outside of the United States for a little while. And unfortunately for him, when he did his postdoc, he had to follow me here. So he did not have the luxury of choosing to do an international postdoc. But yeah, it would be really cool to, get out and see other places and experience that. But another thing I'm considering is doing science writing. And so I know there are a lot of science writing programs across the country, or quite a few at least, where I could expand and get into maybe science journalism or writing for the public.
Well, and that's why you're here on TWiV. You responded. We read your letter last week. You responded to our call for any interested female virologists, and here you are, because you're interested in the communications angle and you wanted to get some experience. So here you are on TWiV. So we hope you like it.
Yeah, thanks for having me.
Yeah. We have a couple of little things to chat about here. The first one was sent by... first let me tell you about the ASM Hot Topics in Microbiology Education online course. It's kind of interesting, a new thing. Help you measure whether your students are truly learning in your classroom or in your laboratory. I don't know, maybe you don't want to really know, but you can. You can design a research project, learn how to gather and evaluate data to discover if your methods improve student learning. Join an online community of supportive faculty, mentors, and facilitators who will work with you through productive interactive assignments. This is all done online. It's a webinar. They're going to take place from August through December of this year, 2017. The registration deadline is coming up August 28th, 10 days from recording now. Go to asm.org/courses. There is a specific link, but it's a long URL. So if you're interested, you might be able to find it at asm.org/courses. If not, we will put a link in the show notes, which you can find at microbe.tv/TWiV, latest episode. Now, Steve, who is not a wet biologist, let's say, he's a computer scientist, he sent us an interesting paper on viral DNA, the type that makes you sick. Well, sort of. Hello, my industrious TWiV masters. The link that you find below is a paper from University of Washington, which discusses the creation of an actual code written in the language of nucleotides for a computer virus dressed up and disguised as a DNA molecule. If this whole concept doesn't mess with your head, then you are obviously not thinking deeply enough. And he sends a link to a paper which we'll chat about. The fortunate thing, my guess, is it probably does not replicate well in vivo, but then your DNA sequencer may not exactly appreciate your playing with such sequences anyway. Thank goodness that DNA sequencers have not yet become self-aware, as they're seeing their masters simply trying to shut them down via a set of deliberate DNA sequences. They might have rogue thoughts with dire consequences to your lab, reminiscent of Skynet. Please tell me your DNA sequencers have not yet incorporated AI as to aid them in performing their workflow just yet. I'm just a computer scientist, but when I saw this, I thought you could use a laugh or two. Keep up the great podcast. Keep your sequencers running safe. This reminds me of Bones in Star Trek. I'm just a doctor. I'm just a computer scientist. Now, I find this a really kind of interesting paper. It is from the University of Washington Computer Science Group. The title is Computer Security, Privacy, and DNA Sequencing, Compromising Computers with Synthesized DNA, Privacy Leaks, and More. And the authors are Peter Ney, Carl Kosherly Organic, Luis Saez, and Tadayoshi Kono. And the idea here is, everyone knows that when you do sequencing nowadays, next generation sequences, high throughput, deep sequences, 454, last episode number, you have to use computational tools to analyze the sequences, to throw out the garbage, to piece them together, because you do short pieces and piece them together and stuff. And so there are a lot of bioinformatic tools. And the thesis of this paper is that, they say this in the first sentence, these tools have encountered little adversarial pressure. In other words, they're not like browsers or email programs or any programs that are constantly being hacked and attacked online. So there's little defense against it. So they wanted to say, if we put an exploit in a DNA sequence and have a machine sequence it and then apply one of the several programs that you apply to such sequences post-sequencing, could that be executed? And so that's what they did here. They actually took a short exploit, which is designed to cause a buffer overflow in a computer, and that will give you complete access to the computer. And they did this by writing this little code. This code probably existed before, and they converted it to DNA using a 2-bit conversion. Like A is 00, C is 01, G is 10, and T is 11, 10 and 11. So they start with this short bit of code, and the idea would be that they made the DNA, they had it synthesized as an oligo, they then put it into the sequencer, they used an Illumina sequencer, the sequencer made the data, then they gave this data to one of the programs that's used to compress the file. So the files that come out of the sequencer are called FASTQ files, and there is a program called FQZ-comp, which would compress it, and they gave it to that program. Now, they did modify this FQZ-comp to make it vulnerable to this exploit.
Yes, that's an important point.
Right, it is. And the program read the sequence, and it immediately called back to the server and gave them arbitrary remote code execution via a shell. So basically, it gave them an exploit that they were looking for.
Keys to the kingdom.
Yeah.
Yes.
So it's a proof of concept, and the reason I think it's interesting is because I think it's true that, the people who are writing these programs don't really think about security and so forth. And so maybe it's time that we start to think about, protecting them against attackers of various sorts. So that's the point of this whole thing here. So
yes,
I think it's cool. Software security.
And despite the fact that they did have to, they introduced a deliberate vulnerability specifically to exploit it with this code, but their underlying point is, look, we've got these programs that nobody's thinking about security for.
Yeah, that's right.
And they are receiving data through a source that we're thinking of as inherently safe, but that's not necessarily true.
That's right. And,
pretty creepy, actually.
Because you think about it, you store these data in big repositories, right? And lots of people have access to them. So this could spread substantially if it were malicious, right?
Right.
So I think it's good that they're raising this issue. Maybe people will think about it.
As a matter of fact, one of my reactions to this paper is that it's a dual use issue. Should they have actually published this? Because this gives somebody the capability of doing this, right?
Mm-hmm.
If you are running the software with a deliberately engineered exploitability in it, which you wouldn't be. But that doesn't mean that there aren't other exploits that could be pulled this way.
Yeah, you typically, if in the real world, if you find an exploit in a program, you first notify the company that produced it, and often they don't respond. You'll tell Microsoft or Apple or whoever, and then after so much time, you release it. And a lot of people say, oh, that's not good to release it. But the point is, if you release it, then maybe the company will do something about it and fix the problem, so forth. So in this case, a lot of these programs are just made by computational biologists. It's the right word, right, Rich?
Yep.
And, maybe they don't want to deal with that. I just don't know how it works. But anyways, anyone's out there that is closer to this, let us know your thoughts on this. I thought that was pretty cool. All right, next we have a snippet, which is also a cool paper. And this is something that we mentioned way back in April when Paul Benash was on the show. And this is all about using pigs for sources of organs for transplant. So the overall picture here is that, we need lots of organs for transplant. If you need a liver or a heart, you get on a waiting list and you can wait a long time. You could even die before you get your organ. So what's the solution? People are thinking about pigs.
Before we get into the discussion of this very, very cool science, it is worth putting this in context, which is that the big reason for the shortage of organs is a shortage of people who've listed themselves as organ donors. And there is a social solution to this problem, which is making organ donation opt-out instead of opt-in.
Yeah.
Unfortunately, that doesn't seem to be in serious discussion anywhere. And as a result, we still have the organ shortage. But if this was, most people are actually okay with the organ donation, but they just don't get around to getting it listed on their driver's license or whatever the procedure is in the state where they live, and the procedures are inconsistent. If that was just the default, that you are presumed to be an organ donor, unless you've opted out, it would still allow people who have some objection to that to avoid being organ donors. But the rest of us, the majority, would just by default be considered organ donors, and anybody dies suddenly, or maybe even not so suddenly, if your organs are usable, they get sent to people who need them, and that would go a very long way toward sopping up the demand for hearts and livers and such.
Do we know that would that take care of it, or
it wouldn't necessarily completely take care of it, but it would be a huge boost.
Yeah.
Is there anywhere in the world where that's done, Alan? Do you know?
I don't know. I think I feel like I've heard of a couple of places that have at least discussed it, but maybe some of our listeners can write in and then mention if they're aware of this being implemented anywhere. But anyway, since that does not seem to be happening, and there are concerns with that. I mean, when you take a human organ, you obviously have whatever that human's history is to deal with, and issues of size. If you have a 1-month-old baby that trying to put an adult liver into that kid is not going to work so well, and vice versa. But so the idea is, well, pig organs are very, very similar to human organs, and
not mine.
Yes, actually,
in size, particularly.
Yeah, in size in particular, and we already raise millions and millions of pigs. So we got this supply there. The two drawbacks to this are obviously immunity, autoimmunity against a donated pig organ, which obviously is going to be very immunologically different from a human organ. But organ recipients are routinely immunosuppressed anyway. So you can get around that. The other issue is what we're going to talk about now, which is that pigs have these porcine endogenous retroviruses, or PERVs, throughout their genome, and there's a tremendous amount of concern that these could run rampant in humans if we started transplanting pig organs into people.
PERV's an acronym that was made for playing with, right?
Yes.
Yeah.
So we mentioned this when B. Nash was around, because he was talking about endogenous retroviruses, and this came up. Could you knock out all the PERVs in a pig cell line? He said, "No, it couldn't be done." We found a paper of George Church, or George was on it, showing that you could, in a pig cell line, knock out like 60 plus endogenous retroviruses, and which is amazing. But using CRISPR, you can do that. And this paper,
I think it was the same group, right?
I think it's the same group, yeah. And this paper will go a little further and make actual pigs lacking.
I have to say that, of course, this caught my attention when it was in the news, and I looked through it casually then. But having now looked closely at the paper, it's technologically incredible.
Mm-hmm.
Yes,
there's nothing technologically new here, really. And, all the components are there, and they've probably been put together. The components have we've made engineered pigs before, but this is my first awareness of it in depth. And this in itself, I think, is an astonishing use of the technology, and the implications for engineering in general are profound to me.
This is an iPhone type of project. No new technology. When the iPhone was first introduced, it had absolutely nothing new in it, and yet it was a revolutionary product because it combined all these existing things in a way that was just really, what the market needed. And this is that. There's nothing new about CRISPR-Cas9. There's nothing new about the procedures they're using, but they're combining all of this, and knocking out all of the PERVs in pigs has huge implications for xenotransplantation.
Every one of the individual steps is a technological marvel all on its own. It's just one mind blower after another, all put together. It's incredible.
So the paper is called Inactivation of Porcine Endogenous Retrovirus in Pigs Using CRISPR-Cas9, and it comes from a large group. First author is Dong Yu. Last author is Luhan Yang, and George Church is on this. Places like eGenesis, a company in Cambridge, Zhejiang University, Yunnan Agricultural University, Aarhus University in Denmark, Third Military Medical University in China, Harvard Medical School, Xinjiang Technology Company, and the Wyss Institute from Harvard. And so the idea is, as we have said, to get rid of the PERVs, not just in a pig cell line, but in actual pigs. And they had previously knocked out 62 PERVs. Remember, these are DNA copies of retroviruses. Retroviruses have previously infected pigs. The RNA genome's converted to DNA. It integrates into the cell. And if the cell is a germ cell, then that will be passed on to offspring, so they become endogenous retroviruses. So over the years, the pig has accumulated 62 copies of pig endogenous retroviruses. And so they use CRISPR-Cas9 to take them all out. And basically, you can design oligonucleotides that will give rise to what we call guide RNAs. And you can pick a conserved part of each PERV so that you put in, say, 2 guide RNAs, along with Cas9, the nuclease that will cut the DNA, and you can cut them all at once and get rid of them all at once. Now, in this paper, they want to do it with primary pig cells, not immortalized pig cell lines, because then they could use those to make actual pigs. And they do that again by CRISPR. They have fewer copies. There's something like 25 to 30 or so copies of PERVs, and they can get rid of them all. They had some problems getting rid of them, which was very interesting. It turns out that when you cut the DNA with Cas9, specifically in these PERVs, it induces programmed cell death, apoptosis. So they had to use an inhibitor of that process in order to get 100% PERV inactivated cells, but they did it. And they showed
basically the cells are freaking out because their DNA has been messed with big time, and they say, "Uh-oh, something bad's going on. We're going to have to kill ourselves to save our buddies, and to save the organism." Okay, so they have to short circuit that.
And then go ahead.
When they first start talking about the cells and detecting 25 copies, they say 25 copies of functional PERVs, and that gave me a bit of pause, because then I thought, well, what are they going to do if there's fragments that might someday recombine or something? But they said that when they did whole genome sequencing, they detected 1 additional copy of a truncated PERV. And so I guess the implication is that they didn't find any other pieces of endogenous retroviruses that could create a problem. That's my assumption of it.
Yeah.
Yeah, and also notable in this, as Kathy already said, is that these PERVs are active. They're producing virus.
That's right. Yeah, if you culture them with human cells, they will infect the human cells, in fact.
Yeah, that's the first part of this paper, is showing, people have been worried about this as a problem in transplantation, is to show that, yeah, indeed, if you culture this with human cells, the human cells will become infected. The PERVs will integrate, and I think that's one of the big deals, is that these are retroviruses. They can integrate into the genome, and that's potentially a mutagenic event. And they take those cultured human cells that now have the PERVs and show that they will actually amplify over a period of time. You'll get more integrations and infect other human cells. So that's a proof of principle that the PERVs could, in fact, be a problem.
So I actually had a question about that. So they show that human cells can increase the copy numbers over time, but in the porcine cells, do they also have copy number increases as the cells are growing? Because then that would indicate that they really need to have 100% of the PERVs knocked out, so that way they're not reinfecting the pig cells while they're growing too.
You would think so, because if they're making viruses, right, those could infect again and increase the copy number, or they could duplicate in the cells. So I would think so, but I don't know. I don't know the answer.
And I'm also wondering if they happen at the same rate as they do in the human cells. Like maybe there's something in the porcine cells that's holding back. Yeah, maybe. You never know.
Yeah, I mean, I would not be at all surprised if here's a project for you. Drop everything. Try this.
Michael, I have a new way to go.
I wouldn't be at all surprised as if the pigs have defenses against these things that keep it in check. And one of the reasons that they can amplify in the human cells is the human cells are probably not adapted for that.
Well, they would almost have to, because the pig genome is stable enough that you can make more pigs that look like pigs, and they survive. And that wouldn't necessarily be the case if you had these retroviruses just still propagating themselves willy-nilly and making additional mutagenic copies throughout the genome. So there's something that's got to be going on there, right?
Yeah.
It's interesting that the PERVs are all, at least in these primary cells, they're functional, which implies they're making virus, right? That's very different from human endogenous retroviruses, where they're not making infectious virus.
Mm-hmm.
I wonder why it's different.
Just as a side note on that, one of the technological things that blows my mind is that, just almost as a side note, one little thing, figure 1b. Oh yeah, we map these things on the genome.
Yeah.
Okay. So without going into detail, there are methods that actually are quite straightforward for figuring out what the integration sites are. And there's a pig genome sequence available.
Sure.
So in a fairly straightforward series of experiments, you can map all 25 of these on the genome. A decade, 2 decades ago, that would have been unthinkable.
No way, yeah. So we should point out that this is never, people have received pig parts before, right? I think valves, heart valves in particular, can come, but there's no description of a perv infecting humans.
Right. I was thinking about this. I don't know about all procedures, but in the cases of heart valves, it's not living tissue really.
Right.
It's that's just cartilage. It's been decellularized. So you don't have an immunology problem. You don't have a biology problem. So I don't know whether there's any cases of using living pig tissue in a transplant.
And the other thing is, of course, if you eventually made a colony of pigs lacking pervs, you'd have to make sure they kept free of pervs, right?
Right.
Mm-hmm.
You got to segregate them away from other pigs or wherever the source of these pervs are, and who knows? So it's not straightforward.
Actually, it'd be interesting to find out if these piglets that they made can catch pervs from other pigs.
Mm-hmm.
Would be.
Yeah. So many peas. Oh, that could be a title, couldn't it?
So many peas.
All right, so then you want to make pigs. And here we take a page from Dolly the sheep. We take out the nucleus from these primary pig cells from which all the pervs have been removed. And then you replace the nucleus of a pig egg cell. Put that into a female sheep, not a female sheep, a female pig, and there you go. You have new pigs born that don't have pervs in them. They can make lots of piglets, and they don't have pigs. They've produced 37 perv inactivated piglets from 17 sows. That
procedure, by the way, the somatic cell nuclear transfer, does not work equally well in all animals.
Mm-hmm.
And there was a whole additional raft of work that had to be done after Dolly the sheep was demonstrated. It turned out sheep were relatively easy to get that to work in. Pigs, I think, were a little bit more difficult, and it was a different set of techniques.
They say they needed to do between 200 and 300 embryo transfers per sow.
Yeah.
That's a lot.
Yeah.
And of those 37 piglets, they still have 15 that are alive, and they're up to 4 months old. So you wonder why those others died, And I'm curious as to whether there's any negative effect of removing all the pervs.
Have they taken these pigs to the opera?
Right.
Well, of course, a major concern in this kind of technology that they address, at least in part in this, is whether when you do the CRISPR-Cas9 thing, there's going to be collateral damage.
Yes.
And they, in addressing that, they noticed that in a couple of knockout cell lines they created, there were chromosomal rearrangements. So they threw those out, and beyond that, they just they sequenced all of the knockouts, because they're knocking out the polymerase gene. Mm-hmm. They're targeting the polymerase gene, and it gets cut and then inappropriately religated by the cells, and that creates a deletion in the polymerase gene that inactivates it. But those can be, random size. They sequenced all of them and showed that the flanking sequences were still integrated in the right place. And yes, all the polymerases were knocked out. And they conclude from that at least within those limits, there are there's no collateral damage. But man, if you're just looking at chromosome translocations and whether or not the pervs are all knocked out right, that's a pretty small sampling of what else could go wrong with the genome. So the real test is, taking the pigs to the opera.
But it could be that a PERV is needed for some, I don't know, liver or heart function, right?
Actually,
you have to know, because you don't want to transplant it into people and then it fails because there's a missing PERV. So
sure.
You really need to study these pigs for a while.
Well, and there's another issue. Another issue is, how are you going to produce them? You need to know that these animals can reproduce themselves sustainably and that you can maintain a population of them.
Oh, yeah. You don't want to be doing nuclear
No,
transfers.
you don't want to be doing SCNT every single time.
For sure.
That's what I was going to ask. Did they say whether or not they're fertile? Have they reproduced? I don't think they said.
I didn't see it.
They're too young at this point
to
even try.
So.
Yeah, they're only 4 months old.
Pigs start pretty early, but 4 months may be a little too early even for them.
Yeah. Anyway, really interesting stuff.
So the other thing I wanted to point out, I said this was technologies coming together, is that the actual first nuclear transplantation experiment was from 1958.
Wow.
Gurdon did it in frogs.
Right.
And I learned this when I was, in college, maybe even in high school. I don't know. But his point then was to prove that every cell contained all of the genetic material necessary for making a complete organism. And so he took a skin cell, I think, from a frog and took the nucleus out and transplanted it into an oocyte and got new frogs. That was a very big deal.
Yes.
But so that's, 60 years old?
But then it was decades before anybody could get that to work in a mammal.
Dolly was 1996.
Yep.
So I want to know, what's the minimum number of genes that you would need to make a new organism? Like a pig or a frog or a sheep. Do you need it all? Or could you put 100 genes or 200 or 500 or 1,000? Somebody's going to do that one day, I'm sure.
Yes.
Yeah. I did find out the sexual maturity is reached by male and female pigs at 5 to 6 months of age.
Okay.
From 5 to 6 months to 18 months is called adolescence for the pigs.
Adolescence.
Okay, so these are tween pigs that they've got right now, and they'll be able to breed them hopefully pretty soon.
Yep. This episode is brought to you by the Defense Threat Reduction Agency. Imagine an everyday, inexpensive drone you could buy online, modified by terrorists to spread chemical or biological weapons over a crowded football stadium or holiday parade. Plague, VX, sarin, weaponized flu. How would we treat the victims? What could we do to counteract the effects? How can we prevent a scenario like this from happening? Join us in Long Beach, California, November 28th through 30th for the 2017 Chemical and Biological Defense Science and Technology Conference to exchange information on the latest and most dynamic developments for countering chemical and biological weapons of mass destruction. Collaborate with more than 1,500 scientists, subject matter experts, military service members, industry partners, and academic leaders from across the globe who are committed to making the world safer by confronting chemical biodefense challenges. Part of the U.S. Department of Defense and charged with safeguarding our warfighters in our nation, the Defense Threat Reduction Agency's Chemical and Biological Technologies Department hosts this important conference and brings together the best and brightest from around the world. Please join us to share your important ideas. For more information and to register, you can go to cbdstconference.com. That's all one word, cbdstconference.com. Stay up to date with the latest conference information on Facebook. You can search for DODDTRA or just go to facebook.com/DODDTRA or on Twitter, CBDST Conference. The 2017 CBDST Conference, today's innovation, tomorrow's warfighter capabilities. We thank the Defense Threat Reduction Agency for their support of TWiV. All right, we have a paper for you, which is possibly part of an arc. We'll see you next time.
Yes.
Can I tell why I think it's exciting?
You bet.
Okay. So the title of the paper is Severe Viral Respiratory Infections in Children with IFIH1 Loss of Function Mutations. And in the big picture is that these authors have identified a human gene which makes individuals susceptible to some respiratory viruses. And that's really cool. And the way that they do it is really cool. They hypothesized that children who are otherwise normally healthy, have no other known diseases, but who end up in pediatric intensive care as infants up to about, I think, 2 years of age, may have something genetically wrong with them that's something that was previously unknown. And so to figure this out, they don't do whole genome sequencing of all these individuals. They just do a shorthand way of sequencing, and they just sequence the exome, which is all of the exons, the coding regions that make proteins. And so by doing that, they do identify a specific gene in these individuals. And so the exome represents about 1% of the genome, but about 85% of the disease-causing mutations are found in the exome. So to me, I've always been interested in host genetics of susceptibility, and exome sequencing has been around since around 2009, and this is just a really nice application of it, I think.
So it's published in PNAS, big collaborative group. First author Samira Asghari, last author Jacques Fellet, and from many places all over the world. And notably, this paper for PNAS was edited by Jean-Laurent Casanova, who is a specialist in this sort of analysis, and we've talked about his work previously on TWiV, for example, looking at why herpes simplex causes severe encephalitis in a certain fraction of individuals. What are the mutations associated with that?
And I'll bet you a large fraction of listeners can identify with this. I mean, I have known a couple of people whose children wound up in the hospital at a very young age with a really, really life-threatening respiratory infection. I'm not saying that all of these are like this, but all kids run through all of these viruses at some point, and a small fraction of those kids have a big problem with it. And it could be that this is quite often the reason. They got something wrong.
These are viruses like respiratory syncytial virus and rhinoviruses and adenoviruses, and we're not talking about like somebody who's dying of flu. These are viruses that we broadly consider pretty innocuous, and the overwhelming majority of the population gets them. They get some sniffles, they feel down a little bit, and then they're over them. But these can send a small portion of the population into the ICU, and so it is a really big deal for the people who are affected by it.
Yep. Respiratory tract infections caused by viruses. Almost 100% of children are infected by the age of 5, and about 3% in each birth cohort get more severe disease, which requires hospitalization. Now, aside from, we don't know any viral differences that are responsible for this. So what else could be responsible? Risk factors like socioeconomic, environmental influences, preterm birth, chronic disease, immunosuppression. But what about, and if you don't have any of these, what else is there? As Kathy has pointed out, then you look at human genetic variation. And this is what we can do now because we have the technology, and it's been done for a number of more severe illnesses. And we talked about one a long time ago on TWiV. It was a study of a single child who had life-threatening influenza, and there were changes in the transcription factor IRF7.
Right.
And what episode was that?
336. And that was patient P.
That's right. Brought to you by the letters
H, N, P, and I.
Something like that, yeah.
And that was a case where the one mutation seemed to be directly related to this drastically worse outcome for flu. Flu, of course, can have horrible outcomes for people with any genetic background, but this particular case seemed to link to that.
Right. So what they did here, they enrolled in this study 120 previously healthy children who had been admitted to pediatric ICUs with respiratory failure because of a viral respiratory infection. And the median age, 78 days. Most common pathogen, respiratory syncytial virus, 56% of the cases, and then human rhinovirus in 26%. And as Kathy said, they did exome sequencing.
Which is a technical tour de force. And if you look at the methods, they basically say, we did exome sequencing.
Yeah.
Yes.
Like 2 sentences or something like that.
No longer worthy of special note.
Right.
No, we had our son's exome sequenced a couple of years ago when we were looking for a genetic cause. And, you go to your doc and they say, yeah, let's sequence his exome. But it's not cheap. And it cost him, we didn't pay for it. And that's thanks to healthcare. It was something like
By the way,
$20,000. Yes.
By the way, just briefly, the way this is done is to take whole DNA and fragment it and hybridize it to oligonucleotides that have affinity tags on them, where the oligonucleotides are a sample of the whole coding sequence, and then use those affinity tagged oligos hybridized to the DNA to drag out the DNA on beads and then sequence the DNA. That's one way. That's the most common way at this point. And you can get a kit to do this.
And then, of course, you do rapid sequencing, deep sequencing, and you analyze the data with programs which could be compromised.
Could potentially be compromised, yes.
So, and all these data are stored in repositories, as we said, they could be a problem. Anyway, that's the other paper. So they found in these sequences from these individuals, they found 6 putative loss of function changes. And this included a change in a protein called IFIH1. That's the gene called IFIH1. It's an interferon-induced protein, which some of you may know as MDA5. That's the old name that we used to call it. And we've worked on that for years as well. It's a RIG-I-like cytoplasmic sensor of double-stranded RNA. It's important for detecting infecting RNA viruses.
So this thing binds double-stranded RNA when it sees it, and that sets off an ATPase activity and exposes a couple of other domains on the protein that interact with other components on a signaling pathway and winds up inducing interferon.
It's a smoke alarm.
It's a smoke alarm.
I have to say that these changes, you can look in a database to see frequencies of different mutations in the population. The database is called the Genome Aggregation Database, or the GNOME-AD, the GNOME Allen Dove.
Oh, nice.
You like that? It's good for you. Anyway, 8 people in this study had possible loss of function changes in this gene, IFIH1. And some of them had splicing variants that would make altered proteins. They had a couple of splicing variants. They had termination codons, which they call in a funny way. What is the word?
Stop gained single nucleotide variants.
So weird.
Stop gained equals nonsense.
Nonsense.
Codon.
I don't know
nonsense mutation, right?
Why don't they use the regular?
I guess because they gained a stop, but that just it seems kind of weird. Yes,
I had to look it up too,
Yeah.
and among other things, I discovered that these guys didn't invent the term.
Right.
It's been around. Others have used it. I don't like it, but I'm an old fogey.
Well, there's nothing we can do.
In some ways, it makes more sense than nonsense. Nonsense means something to us because we've been around for a while. But in a way, it makes more sense to say you gained a stop.
Well, the people who make these terms, I bet you ask them what an amber codon is, they probably wouldn't know.
Right.
Erin, you know what an amber codon is?
I do know what an amber codon is.
Wonderful.
They still teach it in the undergrad classes. So we're good there.
How about, Erin, do you like the word interrogate? Like interrogate the database?
I have no problem with it.
Okay.
I wouldn't say that I
we
love
complained about that a while ago too. We interrogated the data for this. Now, the problem is when a term gets into literature, there's nothing you can do about it because people will use it, and we can complain all we want. Anyway, so they found a number of changes. The splicing variants, end up taking amino acids out of the protein that are known. This protein has been extensively studied. MDA5. So we know that taking these parts out is not good for the activity of the protein. They actually looked in blood cells from one patient, showed that one of the putative splice changes did result in a shorter protein as expected. And so they can show that each of these, I think it's 4 changes, would really mess up the protein. They also could get some of the parents and show, they could tell whether the children were homozygous or heterozygous. They have a nice chart here, and then they could, in some cases, they could get the parental DNA and show what the parents had. For example, here, one of the children was, the parents both heterozygous for the mutation, one heterozygous in the father, one heterozygous in the mother. So presumably all of them inherited these changes. Right. Now, the functional characterization is important here. So what do these mutations in the genes encoding MDA5 do? So they do this in cell culture, and they do this in cells that are deficient for these proteins. Well, first they start by simply putting plasmids. Oh yeah, they do use these cells that are deficient for these proteins, right? They take plasmids encoding the wild-type protein, and then they're altered proteins. They have 1, 2, 3, 4 different altered proteins. And they show that the production of the altered proteins inhibits the activity of the wild-type protein. And by the way, the wild-type protein, when you add double-stranded RNA to the cells, it results in the induction of interferon. And that's interfered with by these altered proteins. And in fact, they have other defects. They're less stable. They don't have ATPase activity, which is one of the activities required for induction of interferon. So these changes are clearly disrupting the activities of these proteins. So there's no doubt there that they're not good for the activity of MDA5. The other thing they look at is whether they're important for virus replication. It turns out that 6 of the patients who had a change in the MDA5 protein, they were infected with respiratory syncytial virus, and 2 of them were infected with rhinovirus. So they take these 2 viruses and ask in cultured cells, do these changes in MDA5 make a difference? And they use for these experiments a liver cell line called HUH75, which doesn't produce MDA5 and has an altered form of RIG-I, so they can't react to double-stranded RNA. So you put wild-type MDA5 into these cells, and that inhibits the replication of rhinovirus and respiratory syncytial virus. But if you put in the altered proteins from these patients, they don't inhibit the replication of these viruses. So the proteins are defective that are encoded in these patients' genomes, and there's a problem in their ability to restrict viral replication in culture. So the suggestion here is that maybe these altered IFIH1 alleles were responsible for more severe disease in these patients. And I think they say an independent study was just published showing another change in IFIH1 in another patient infected with rhinovirus. So that's good. What I would have liked to have seen is taking cells from maybe respiratory cells from these patients, culture them, in fact, show that the virus replicates, and then put in the wild-type MDA5 gene and show that complements the defect. Because there's always the possibility that there's something else in addition to the MDA5. But they didn't do that here. Maybe they'll do it next time. Or it's not always easy to get cells from the patients. Remember that flu paper, patient P, that patient had donated a lot of cells, kept going back, and they actually made a point of saying that. And I think they did these kinds of experiments, but they weren't done in this study. So what do you think? Is this just suggestive, or are you happy?
Oh
with
no,
this?
I'm convinced.
You're convinced?
Mm-hmm.
Yeah, absolutely. Of course,
I'm pretty sold on it.
Yeah,
I think it's suggestive, but I'm not convinced because I'd like to see whether this is the only change that's doing this, or is there something else? Yeah. Because you might miss something in the exon sequencing, or it may be subtle, and it may be. I mean, what if you took cells from these patients and you put in wild-type MDA5, but it didn't it wasn't enough to complement the defect. Maybe something else is there as well. I just like some of the other studies do that, and I think that's important.
Mm-hmm.
Well, I think I was convinced by the experiment at the end where they used the MEFs that were MDA5, IFIH1 minus, and then they put in the gene and got the expected result if it was causative. So
right, but those are not from the patients, of course.
No.
And the patients may have other changes. That's what I'm saying. That other changes may be part of this. It may not simply be IFIH.
It could be multigenic, but this is certainly probably a major gene.
I would say.
I suppose one way that this suggests that this is the only thing or the primary thing is that you've got 7 or is it 8 patients who are genetically probably completely different, and this is the one thing they have in common.
Right.
Right.
Yeah, I think the independent study is also good, and the more if you do this over and over and you get similar things, I think that's fine. But so far, I think the N is not big enough to convince me.
I was wondering. Oh, go ahead.
A subtlety here that I don't. I was cruising on this subtlety, so I don't know if you got to this. Did you discuss the dominant negative aspect
I
of this?
didn't do that. Go ahead. You can.
So what they find is that if you express a mutant form of this protein along with the wild type, first of all, that the mutant forms are less stable than the wild type. That's not an uncommon thing for a mutant protein to be turned over. But they find that if you express the mutant form along with the wild type, it actually causes a faster turnover of the wild type protein as well, and compromises the function of the wild type protein. This is what is called dominant negative. So you have a bad thing and you mix it with a good thing and you get the bad thing result. And the implication from that usually is that this thing functions as some sort of multimer, so that the bad subunit poisons the good subunit. And so I've been looking up to see if I could find evidence for that. And yes, indeed, there is evidence that MDIE5 at least forms dimers, and when it complexes with RNA, forms multimeric filaments, in fact.
Yeah.
So
and that
it's
also
easy to
fits
see.
with also fits with the genetics that most of the patients were heterozygous for this.
Heterozygous, yes. Good point.
Had the phenotype.
So that's that's actually the ATP dependent reaction. The polymerization, the recognition of RNA leads to polymerization of MDA5 into a filament.
Mm-hmm.
And that's important for the next step. So that's probably where that's happening.
I was wondering, what happens to these kids after they get over this medical crisis? Do they develop sufficient immunity to these viruses that this doesn't happen again?
They talked about that a little in the paper, and most of these kids have no other problems.
Yeah, they're fine.
Yeah, so
or have had no other problems. There was one child, I think, that had repeated hospitalizations for respiratory infections, but most of them, the rest of their medical history was uneventful, unremarkable.
Right, but, so what I'm wondering, how does that work?
Yeah.
Well, this is just one part of the immune response. So if you can get past this, ultimately, to clear the infection, I would assume you have to mount a secondary immune response, and that helps you in the future.
Right. Or maybe there are a lot more people with these mutations walking around, and we're only seeing the ones who had the relatively rare bad outcome.
Could be.
Maybe. There are other people with severe respiratory infections that we haven't looked at, so that may be part of it. And also, what's puzzling is that with rhinovirus, at least, you're going to get, there are hundreds of different genotypes that aren't cross-reactive in terms of the immune response, right? So you would expect them to be infected every year, right? But why they don't get severe disease every time is weird, right?
There could be some part of the immune system that's developing when they're that young, and then as they get older, that goes away.
Right.
Yeah, or that it's just stochastic. That they happen to get this particular infection, and they have this genetic mutation, and all the stars aligned, in a bad way. And the next time they still have that gene, but the next time they get the infection, maybe, as somebody had already said, some other aspect of the immune response.
And also that
took precedence.
It could depend on dose, right, of virus. It could depend on nutritional status, or all sorts of things. Yeah, could be. And the same with RSV. RSV does not give such robust, long-lasting immunity, right? It's the mucosal immunity, which is important, wanes pretty quickly. So at least early in life, you tend to get multiple infections. So there must be other things going on.
Yeah. So these really severe respiratory infections seem to happen in young infants, and I wonder if that's because this is the first time that they've seen these pathogens, or if it has more to do with the fact that their immune system is still maturing to some extent.
Right, like Erin was just...
hard to know.
Right.
Would be nice if they had some serum as well, so they could see if there was a previous infection,
Right.
But we didn't. Now,
they had blood samples from them, so I would assume that they could have
looked at antibodies,
taken some serum. Yeah.
It's not here. Yeah. So the question is, what do we do about this? So presumably, at some point, everyone's genome will be sequenced. We'll have a whole catalog of mutations in various immune genes that are associated with severe infections of different sorts. What do we do with that information?
Is it useful to know this about your kid?
Yeah, you can't do anything at the moment, right?
What do you just do? Sit around being nervous every time they sniffle?
Right. I mean, you should get if there are vaccines available, you should get them anyway, right?
Right.
Oh, that's the other thing they noticed, is that none of these kids had any problem tolerating vaccinations, including the live vaccinations. Yeah. That, to me, is interesting. Important, too.
So at this moment, we don't know what to do with this information.
Right.
Someday we may have rhinovirus vaccines, we may have respiratory syncytial vaccines, and those are the two big ones they found here. So this information, maybe you would have this information, and either way, the doc is going to say get immunized, right?
Yeah.
Whether you have
right.
changes.
But this could also get us to the kind of mechanistic insight that we need to be able to do a better job managing these patients. They end up in the PICU, and if they're well cared for in a highly advanced PICU, then their outcomes tend to be pretty good. After all the risks of being on ventilators and what have you. But if we could somehow intercept this earlier, here's a kid, he's developing a severe RSV. He's got the IFIH1 allele. Let's use, whatever intervention would activate their immune response against the virus.
Yeah. Yeah, I mean, we don't know the answer. It's an interesting thing to consider, right? So this is theoretically very interesting.
Yes.
Right. In fact, the way they say it is, it's an immediate practical implication that there's the possibility to develop diagnostic assays, but then the specific preventive and interventional measures is kind of what we don't have at this point necessarily. So.
Yeah, and a diagnostic without an intervention, there's a lot of debate about whether that's even a good thing to have.
Yeah.
Do you want to know? Is it if it's not going to help you?
Right.
These are the big ethical questions going into the genetic era here, as you said.
Yes.
Yep.
It's within a decade. Genome sequence is going to be a common part of everybody's medical record.
Right.
And, we're in Gattaca, right?
Yep, we are.
And so the ethical questions are profound. We're going to have to deal with them.
Yeah, but it's great. It's great that we have to.
Mm-hmm.
Mm-hmm.
Love it.
Yes.
Just love it. Any thoughts, Erin?
You guys are basically covering all the thoughts that I've had.
You going to have your genome sequenced, Erin?
I would probably not have my genome sequenced. I'm kind of the opinion that I'd like to not really know.
Yeah.
Oh gosh, millennials are going to kill the genome sequencing industry.
There are multiple people in my lab who have had their genome sequenced,
right.
And there's actually a postdoc who really likes to pull out his genome every once in a while and be like, look, I am immune to this thing and I'm immune to this thing. So I don't think all of us are going to kill it.
Yeah, I haven't, but I did do the 23andMe years ago, and it's kind of interesting to see that. But I would, if it weren't so expensive, I'd have it done. The whole exome, I guess you could do. But
did you say your son's was 20,000?
Like 20 grand, yeah.
But that was a few years ago, wasn't it?
Yeah, so that was 3 years ago, but
okay, so it's probably 10 grand now.
It shouldn't have been so much. But of course, the insurance paid for most of it, but it's just shocking because it can be done for much less, right?
Yes.
And I would love to get a hold of it and look to see if there's any of these, IRF7 or IFI H1, etc. I don't know how to do that. I
definitely think this stuff has its utility, but I have a personality where I worry too much. So I would see things and panic.
I would love to have my sequence and then pull out every day a gene or two and look at it and say, oh, this is cool. There's nothing here. Okay, let's move on. I would love to be able to do that. But I guess I could do it in my own lab. But all right, let's do a few email here. First one's from John. Dear Vincent, I started listening to the TWiX series of podcasts about 4 months ago, and I wanted to write in and say a big thank you for all the work that you and the rest of the Tweemsters have put into TWiX. Currently working as a consumer healthcare rep and have had my days livened up by fantastic discussions of news, studies, and clinicals. I was blessed to begin my career in the Vilas Boas lab at the School of Biological Sciences at the University of Auckland from the age of 19. Check out the lab if you get a chance. Dr. Silas Vilas Boas leads a metabolomics group with an emphasis on fungal secondary metabolites, among other areas. I'd highly recommend his lab to anyone with an interest in pursuing microbial metabolomics at a graduate or postgrad level. He's a great supervisor with lots of industry knowledge, focus, and contacts. Life intervened in such a way that I lost my passion for science toward the ends of my bachelor's in biology statistics, emphasis on microbiology. Long story short, I encountered difficulties in reconciling the traditional values I'd been brought up with and the fact that I lived in the 21st century. It's all fine now. It required a few years to understand, but life is much better now. Recently, that passion has come back in a greater, more mature and sincere way. I must credit the entire TWiX team for this. I started on TWiV, went to TWiM, then TWiP, then TWiVo, and now I don't know how I will be able to catch up. I don't know where my career will go from here, but I can say you all helped rekindle a love I thought long gone. Sorry for the long-winded email, and thank you for all your hard work. TWiX is as viral as ever. May your exponential growth phase be forever like that of a continuous chemostat culture. Best regards, John from Melbourne, Australia. P.S. Nearly forgot. I'm sitting in Hong Kong airport where it's a balmy 28°C outside with 79% humidity. I suspect conditions inside the airport are closer to 22°C and very little humidity due to a massive air conditioning effort. Nice. Thank you very much, John.
Thank you.
This sentence here. Long story short, I encountered difficulties in reconciling the traditional values I'd been brought up with and the fact that I lived in the 21st century. Yes. That sounds like a pretty intense struggle.
Mm-hmm.
Yes. Good for you.
Happens. It does happen. Yeah. Rich, can you take the next one?
Sure. Jeffrey writes, Dear TWiV crew, podcasts may not be Facebook, but that doesn't stop you all, other TWiX series included, from being my favorite nerds of trust. And he provides a link to a journal article. Where was this?
PLOS Biology.
PLOS Biology, called Practices and Promises of Facebook for Science Outreach, Becoming a Nerd of Trust. And it's a long article about how Facebook can be used as a science communication platform. Let me go on with the email here. Incidentally, sad to see, though, that controversial topics on Facebook include climate change, vaccines, evolution, genetically modified organisms. Good to see that they aren't so controversial on TWiV. Incidentally, it's fun to see that there's a lauded and quite scientifically minded column and cookbook called The Food Lab, written by none other than Fred Alt's son. A step past Blue Apron, though, unfortunately, you do need to dig up the ingredients yourself. Keep up with the outreach. Jeff from Canada Land.
So I've been saying this for years, that everybody's on Facebook. Many people are on Facebook, and families, friends. So why not use it to put your science there? Because you're going to hit them. And so that's what this article is all about. So it's a formal declaration of same.
It's interesting that part of this article points out something that probably many people have observed, and that is a lot of people seem to use Facebook just for social stuff and Twitter for more professional stuff. You know, they do their families on Facebook, and they do their work on Twitter. Why is that?
Yeah. I don't know. Maybe it evolved that way. Yeah, it is true about Facebook. Yep. Fred Alt. Now, Fred and I were postdocs together many years ago in the Baltimore lab, and then Fred and I both came here to Columbia. So actually, Fred and Steve Goff and I were 3 postdocs there. We all came here to Columbia. Steve and I are still here. Fred went back to Boston. He's at Harvard and has had a huge, illustrious career. And I didn't know this about his son, but it's a cool website, the Food Lab. He's into cooking. Very neat. Alan, can you take Steve's, please?
I can. Steve writes, phages inserted in E. coli genome promote hemorrhage with antibiotics. And he sends a ProMed mail link. Says, hi, Vincent et al. A tragic story from ProMed mail highlights a virus, bacteria, antibiotics interaction that I don't recall hearing about before. Certainly a terrible blow to clinicians trying to do their best to save patients. To find that a logical decision to give antibiotics to sick children can actually set off this dreadful hemorrhagic process. Perhaps you could usefully discuss what is known about the mechanisms behind this in one of your podcasts. And Steve's in Luton, Bedfordshire, England. And what is this?
So
those are quotes from the ProMed Mail article.
So these are strains of E. coli that cause very serious diseases, and they're O157 isolates. They have a plasmid on which a prophage on which there are Shiga toxin genes. All right, it's a lambda-like bacteriophage genome integrated into the genome, and this is what makes them lethal. We've talked about this on TWiV, actually. And it turns out that you cannot treat people with fluoroquinolones because they induce expression of the toxin genes from the prophage, and it makes the disease worse.
So that's the mechanism, induction of the toxin genes. Okay.
That's it. And so you have to diagnose this very early on before you start treating. Otherwise, it makes it worse. Yeah, we've talked about this on TWiV.
So there are other antibiotics that you can use that wouldn't do this.
Yes. Okay. Kathy.
Anthony writes, Joel Clement says the Trump administration is threatening public policy and safety by trying to silence scientists. And he gives a link to a Washington Post article. And Joel Clement was the director of Office of Policy Analysis at the U.S. Interior Department. And he and a number of other people have been removed to other positions in the Interior Department that are not necessarily relevant at all to their skills and training. And
they've been sent to administrative Siberia.
Yeah. Involuntary reassignments. And so the article just describes he describes this whole phenomenon and comes down with how this is really not good for science and for the public, basically.
Yeah.
And it's a story that somehow I had missed. So it was very interesting to read.
It's difficult to keep track of just how much is wrong these days.
Oh, boy.
Yeah.
Oh, boy, is it.
Yeah.
Unbelievable.
This is another in a series of articles that I think of as the sextant was broken aboard the Titanic. It's just... Yeah, this is a problem, and that's a problem, and the other thing's a problem, and it's just all fallen apart.
To me, the biggest problem is that there's too many people who don't think there's a problem.
Yes.
That is very true.
That's the most disturbing thing.
Kathy, you can take the next one as well.
Sure. Ricardo writes, hello, Vincent. Have you seen this? And he gives a link to a film called Unrest And this film came out, I think, in about January of this year. It's a documentary about myalgic encephalomyelitis, or ME, and which was previously known as CFS. And the director has ME, and I didn't figure out how the director was able to have enough good time, as it were, to put the film together. But I think now the film is available. You can just download it maybe and stream it online.
Looks like.
Yeah, I haven't seen it. I should look at it for sure. I was sort of aware that this had come out, but I haven't seen it. So thanks for pointing that out. I will check it out. No one else has seen it, right?
No.
No, I just looked at the site.
Kathy, can you take one more, please?
Sure. Anonymous writes, TWiVirions. I look forward to the upcoming episode from Montana. I hope you have a great trip. I really enjoyed the recent series on antiviral RNAi, especially the comments from Drs. Sullivan and Tanuver. You mentioned that Victor Ambros was not among the 2006 Nobel laureates for RNAi, so I wanted to point out that it would be difficult to justify including him, but not Gary Ruvkun, his longtime collaborator and co-discoverer of microRNAs. They were studying timing in worms, another example of serendipitous fundamental discovery arising from purely curiosity-driven research. Ambros and Ruvkun were awarded the Lasker Prize together with Sir David Balcombe, who will lead the newly announced Cambridge Center for Crop Science, and Anonymous gives a link to that. Speaking of plants and RNAi, listeners who recall the Voynette saga may be interested in a related editorial that accompanies a recently published paper, and he gives a link to a Nature article about an article that was in Nature Plants. In fact, the editorial, I think, is in Nature Plants, and talks about the fact how a former co-author with Voynette has now published something and included all of the raw data as supplemental information. And so that they're saying maybe should be the standard for the future. But obviously, or at least to me, it seemed like this person is really trying to overcome any of the bad things that he was, unfortunately tainted with by being a co-author.
Of course, you can also taint supplementary data, right?
Yeah.
Yeah, releasing the raw data, if there was manipulation of the raw data, doesn't really get you past the problem.
Yeah.
I mean, it's obviously in this case, you've got somebody who was apparently up to some shenanigans and their co-authors weren't aware, and it's a mess for everybody, and now they're trying to salvage what they can out of it. And I understand that and respect the effort, but I don't think there are easy solutions here.
Where does that saga stand now? Is Voynette pressing on?
I haven't followed it very closely. I probably ought to follow it more closely, although I find these types of stories relentlessly depressing. So I don't know what the status of it is.
All right.
I don't either. Maybe someone could update us on it.
Yeah.
But Ben Tanuver made some comments after the show last time, but so he's aware of it as well. But, the subtitle of this article, transparency is the only way to guarantee that research results can be trusted more than some sporting achievements. But that's the problem. Not everyone is transparent.
Right.
So competitive that people don't want to be transparent. Oh, well, let's do one more. Erin, can you take John's, please?
Sure. Ye TWiV. Happy to have a good reason to write in again. The weather in Soho, New York is much the same, I imagine, as it is up at Columbia today. 29 C, sunny. I heard your conversation on this past week's TWiV 452 regarding manganins and their discovery in frogs. This caught my ear as I've known about manganins since long before I even knew what a PhD was, let alone had one. My pediatrician told me about them when I was about 8 years old, specifically because he thought they were interesting and because I speak Hebrew. Manganins were named for the Hebrew word for shield, and I thought you and the TWiV audience would be interested in hearing that. In English, it's usually rendered as magan, So magonins would be more in line with what I'd expect. Magonins would be more in line with what I'd expect, but magonins is the spelling we have. A magon is also used in Magen David, which is often mistranslated as Star of David, but in fact means literally Shield of David. Today, it's more often called the Jewish star. Magen comes from the same Hebrew root that gives us Haganah or defense. The points being that magonins, because of their origin in the word magon, are essentially also named defensins. They just happen to be named that in Hebrew.
Interesting.
The last time I wrote to you, I had just finished my PhD and was on the hunt for a job. Shortly after that, I became a full-time science communicator as a medical writer, helping pharmaceutical companies provide fair and balanced educational and medical science communications regarding their products. It's the sort of career that I was considering when we did TWiV 324, and I must say that I would highly recommend it to any young scientist who wants to stay close to the data, but also wants to be in a communications-related scientific role. At present, I mentor a variety of postdocs and students who are interested in transitioning to the field and would be happy to speak to anyone else you or the rest of the TWiV hosts may know who might be looking for a description of the field. Best and thanks for all that you do, John Schuyler.
John, I would just suggest that you not use the phrase fair and balanced.
It was probably inadvertent.
Yeah, I think it probably was.
John was on TWiV 324.
I'm sure John is aware of the context of fair and balanced. I don't know, maybe it was intended satirically, but...
and knowing John, it probably was.
Yeah.
I don't know, Erin, if you remember, but John used a nom de plume. What do you call it when you use a different name?
Nom de plume.
Pseudonym.
Pen name.
Pseudonym. John Schuyler was his pseudonym because he used to do a lot of writing, I guess, and he didn't want his thesis advisor to find out who he was.
Oh, okay.
And then on TWiV, he revealed himself to be Leland Feynman.
I was wondering why there was a second name there, but
Yes.
I just kind of brushed over that.
A little bit unusual. All right, let's do some picks of the week. And Erin, I see you have something for us.
I do. So, actually, I picked a book that I have given out as a Christmas present before, and it's called What If? Serious Scientific Answers to Absurd Hypothetical Questions. And it's written by the creator of the XKCD webcomic. It's the comic by Randall Munroe that's got stick figures, and they go through all these like physical or physics questions or just about the world in general. And some of the questions that he gives in this book are fantastic. So he gives real scientific answers to things like, what would happen if to the Earth if the sun suddenly switched off?
Right.
So he kind of goes into depth on like, well, we would all die, but if we didn't all die, this is what would happen. And my personal favorite is, if everyone on the planet stayed away from each other for a couple of weeks, wouldn't the common cold be wiped out?
That's good. Does he talk about what would happen if you found out you had a mutation in IFIH1? No.
He does not touch on that, but he does mention that some people might be more susceptible.
It's an amazing website. I just don't know how they do it. It's just him that does this XKCD?
I think it's just him.
It's amazing.
It is just him. Yeah,
So
he
clever.
worked at NASA for a while.
NASA engineer for a while, and then I guess he was doing webcomics on the side, and eventually the webcomics got to be a big enough deal, and he had a big enough following that he quit the NASA job, and this is what he does now.
Amazing. That sounds pretty cool. I'll have to check that out. Alan, what do you have?
I have is something that people may find consoling next week if you are not able to get to the eclipse zone, or if you get there and you find it's totally clouded over, or air traffic control won't give you the clearance you need. Not that I have any stress about that, but I think a lot of people may be in a position where they're saying, oh my God, this thing was hyped to the rafters as the chance of a lifetime. I'm never going to see it again, and I missed it. Don't worry. This is a wonderful Washington Post article / infographic / animation. Try and look at it on the biggest screen you can find. They show upcoming eclipses, and you can plug in your birth year at the top, and they will, based on average lifespans, they'll tell you how many eclipses you can expect in your remaining years. So Let me plug in my birth year. I forgot what the answer was. There are 34 more total solar eclipses worldwide in my lifetime.
We can date ourselves this way, huh?
Yes. So I've got 34 eclipses left. Erin's going to be in the best shape here.
Let's see. Yes, I will have 51 more total solar eclipses.
You're set. You don't have to worry about any of this. But importantly, the next one to hit the U.S. is in 2024. So it's really not that far off. And for a lot of people, it's actually going to be more convenient than this one because it goes from southwest, actually crosses Mexico, goes into southwest Texas. I don't see Austin on this map, but it may actually go over your house, Rich.
No, it does not.
Okay,
we get about a half of an eclipse.
Okay, so you'd have to go
north,
travel just a little bit. You'd have to travel north, but then it goes up across through the northeastern U.S. and leaves in Maine. So
the one in 2024 is April 8th, 2024, and I've already put it on my calendar and put in an alert months before that so that I can make plans.
You can actually get there.
It's going to come pretty close to Ann Arbor, right?
And it's in fact, it's going to go right over the city of Dallas.
There you go. So if I put in my birth date here, it says, take your vitamins. You'll be 76 the next time a total solar eclipse goes over North America. I've only got 22 more possibilities worldwide. And I'm going to have to go to Africa. Oh, well.
I think I can go visit my parents in 2045. It looks like there's going to be one that goes over New Mexico at some point.
There you go.
So I'll have to make sure they're still living there.
Rich, what do you have for us?
Got to shift gears here. Okay. So this is a pick of local interest that comes under the realm of giving a nascent science communicator a TWiV bump. Mm-hmm. There's I guess you'd call it a gallery. She calls it a gallery in Austin that I went to yesterday with my grandchildren called art.science.gallery. This is, it's run by a woman was founded by and run by a woman named J. Haley Gillespie, who lives in Austin, was educated in Austin. I don't know if she's Austin her whole life. She has a PhD in ecology from the University of Texas at Austin, where she studied a threatened salamander that lives locally. And she's also an artist and is interested in the convergence of art and science, and has opened this gallery that she calls the Art Science Gallery in actually a fascinating complex of buildings out in East Austin that's essentially an artist's colony. And she has shows that turn over about every 3 or 4 months in the gallery, where she solicits people to contribute in a given topic, and they contribute their art relative to some aspect of science, and she puts together a show, and it's delightful. The current show is on birds. Okay, so you can go and see birds with various different prints of birds with a scientific bent. Like one was a lot of nice paintings of birds, parrots on a phylogenetic tree, etc. So it was great fun, and I wish her. She's interested in all sorts of science communication, and I wish her the best.
It's cool.
Very cool.
Can I come?
Yeah, absolutely. we could have a TWiV party at the Art Science Gallery. They'll rent it out to you.
Oh, yeah?
Sure.
So it's still going to be around next year.
Yeah. Actually, she just had a fundraising thing. There's a thing here. She had some sort of, let me see. The hashtag Save Art Science Gallery fundraiser. Thanks so much everyone who helped our crowdfunding campaign. We made our $20,000 fundraising goal, and we'll stay open for another year.
That's cool.
Yeah.
Looks like a neat place.
It is a neat place.
I like this space. Looks neat. Kathy, what do you have?
I have a site that's the Astronomy Photographer of the Year shortlist, and it's just got a bunch of really beautiful astronomy photos. Some where they've done multiple exposures and, combined the background with the sky. There's one toward the end where they didn't do that, and it's the picture of the International Space Station across the front of the moon, lit up by the sun in broad daylight, and that's a really cool photo. But they're all just remarkable, I think. Really lovely.
Yeah, they are.
Nice. There's nice captions about them too.
Yeah, and I was really interested that they told you how the photographs were made.
Mm-hmm.
Okay, because it gives you some idea of the difficulties involved.
I love this.
They're beautiful.
Jellyfish nebula.
I was just looking at that one too.
I love these.
Really pretty.
These ones that are so far away that
Mm-hmm.
and they're so weird looking.
Yeah. Or the picture of the aurora up through trees that glow pink because of the street lamps in Russia.
Right.
It's just amazing.
Yeah, I'm glad those aren't cherry blossom trees. Cherry trees.
Oh, there's snow on them. That's why they're pink.
Mm-hmm.
Oh,
they're
okay.
reflecting.
I thought those were leaves, and I was really confused.
Yeah, no.
I have an interesting video this week. It's a YouTube video, which is a speeded up video of a guy taking apart an automatic transmission from a Ford Thunderbird. And the thing that's amazing about it is that the thing is so complicated. There's so many parts, and I just hadn't realized that. I don't really like automatic transmissions. I prefer to drive a manual transmission, but this has so many there's one part where there's this cavity with all these compartments in it, and I just don't even know what that does. And I don't know how he gets it all back together again at the end. He cleans it up and puts it back together.
It occurred to me that if he had to, he could go back and look at some of the photographs that he'd been making in the course of doing
That's right. That's right. Well, whenever I take something apart, the first time I always take some pictures because that way I'll remember how to put it back together again.
Yeah.
That's why having a cell phone nowadays is so great. You can do that.
Well, I watched this whole thing, and I watched a subsequent one that popped up from the same group where they reconditioned an old Ford V8 flathead engine that was just as fascinating. And I have several reactions. First of all, my $30,000 car is cheap. Okay, I can't believe. I can't believe the technology. It's just incredible. And second, the passion of these people and the skill to do this is just amazing. It takes extraordinary skill, I think, and knowledge to take apart an automatic transmission and clean it up and put it back together. And why would you want to do it anyway?
Why would you even do that?
It would
clearly they love it.
Oh, cleaning it out, yeah. I mean, if it's broken, you probably just get a new one, right?
Right.
I don't even know if you take them apart, but it's all about passion. That's what it is. Yeah. You said it.
Yeah. I had a friend rebuild my carburetor on the kitchen table, and it was the same kind of thing. It's like, why would you do this? You can buy a rebuilt one, but no, it's really cool. You get to dip it in all this stuff, gets it all clean, and you know how it works.
Yep.
I'm trying to remember what this group was, because it's a classic cars group.
The YouTube channel?
Haggerty.
Yes.
Haggerty. If you look at Haggerty classic cars, they do all kinds of stuff. And the one with the Ford engine, they've got this incredibly beat up old pickup truck. Okay. And they yank the engine out of it and they recondition it into this thing. It's got a couple of like 2 4-barrel carburetors sitting on the top of it, and it's all shiny and everything else, and stick it back in this old crummy truck and drive it out of the place. That's really fascinating. Great stuff.
The whole thing is it's just something I wouldn't do, but it's cool.
Yeah.
We have a listener pick from Brian. Hello, TWiVirium. Longtime listener, first-time writer. In the suburbs of D.C., it's a sticky 29C. In regards to your recent mentions of predatory journals, I was wondering if you all had seen this story posted to Science Alert. And this is a story of someone who submitted a paper. to 4 dodgy journals, and the paper was on midichlorians, of course, which is a fictional thing from Star Wars.
Star Wars.
And it was accepted by some of them, despite having incomplete paragraphs lifted from Star Wars. Amusing to say the least. Please keep up the good work with the TWiV podcast. I think we've mentioned similar things that have happened in the past where people send in bogus studies and they get accepted because they just want the money.
Yep.
I thought what was interesting about this was that they used some information from the Wikipedia page on mitochondria and reworded it to be midichlorian and makes it sound more authentic.
Right.
But as Vincent said, it's got paragraphs like this one in the middle of it. Did you ever hear the tragedy of Darth Plagueis the Wise?
Yes.
I thought no.
I mean, it clearly wasn't reviewed or even looked at by anyone, right? Yeah. It's just predatory journal. Stay away from them. You basically have to know your field and what journals. We grow up learning what journals to submit to, right? And you just don't go beyond that. There's no need to. All right, that'll do it for TWiV 455. I can't find anything significant about that number. So we'll just leave it at that. You can find it at Apple Podcasts, microbe.tv/TWiV. And if you listen on a mobile device, your cell phone or your tablet, just subscribe to TWiV so you get every episode as they're released. That way you don't have to go. I know a lot of people tell me they go to their computer every week and they play the episode. That's great, but you should subscribe so you automatically get them. That would really help us too. And if you like what we do, consider supporting us financially. Go to microbe.tv/contribute. And there are multiple ways that we have that you can help us out. We'd be eternally grateful for your support. Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Kathy, I just saw this cool video. There's a lady in Ann Arbor who makes tiles. Do you know of her shop?
There's
lots.
Well,
this one is really cool. I have to get the name of it because
send it to me.
It's so cool. She has a way of making these little ridges on the tile, then filling it with stuff that gets baked in it. It's right on the main street in Ann Arbor there, and it looked familiar to me. She's very cool. It's a very cool. I have to send you the link. You would like it.
Yeah.
Rich Condit, formerly and actually an emeritus professor, University of Florida at Gainesville, currently residing in Austin, Texas. Good to have you back, Rich.
Great to be here. Always a good time. Thanks very much.
Alan Dove is at alandove.com. He's on Twitter under the same name. Thank you, Alan.
Thank you. It's always a pleasure.
Our guest today from the Fred Hutchinson Cancer Research Center in Seattle, Washington, Erin Garcia. Thank you, Erin.
Thanks for having me. I had a great time.
I hope you come back sometime.
I would love to.
That's a line from some show. Y'all come back sometime.
Yeah.
It's an old one. I'm Vincent Racaniello. You can find me at virology.ws. And I want to thank the sponsor of this episode, the Defense Threat Reduction Agency. Check out their upcoming conference at cbdstconference.com. The music you hear at the beginning of TWiV is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Hey, how are you?
Good.
Harlan. No, not Harlan.
No.
Wait, just wait. Hang on. I got to think. Is H the right letter?
H is the,
oh, gosh, I'm getting so old. Harlan is a boy's name, I guess.
You want a hint?
If you just give me some time, I might come up with it. Otherwise, I'll feel really stupid. Haley?
No.
Hayden?
No.
Heather?
No, you asked that last time.
Heather?
No.
Harper!
Yes.
The one, Kill a Mockingbird.
Yeah.
You see, if you give me time, eventually I'll get it. The problem is in science, nobody gives you any time. How are you, Harper?
Good.
Did you have a long trip in Oregon?
We didn't go. We were here. But we did go on a couple little vacations.
You didn't go to Oregon?
No, I wanted to, though.
Well, you stayed there with your parents?
Yes.
Huh.
They took care of the house.
Mm-hmm.
I see.
We put origami all over their house, and when they got home, we were hiding, and they didn't know we were here.
It's funny.
So it was a surprise welcome home. That was fun.
Yeah.
Origami.